Thanks again for coming. Benchling has grown tremendously over the last couple of years, and with that, our community of users and customers has grown as well. And so tonight is really about bringing everyone together to share and learn. So with that, I want to turn it over to our first presenter, Jeanette. Jeanette Grant is the Associate Director of Project Management at Adicet Bio, where she's responsible for the oversight of growing a pipeline of candidates and directs the project management department. Prior to Adicet, she was a project manager for research stage programs at Agensis, a mid-sized biotech in Santa Monica focused on antibody-drug conjugate development in oncology. She completed her dissertation research in the lab of Dr. Stephen Dubinet at UCLA in lung cancer carcinogenesis.
Thank you. So again, my name is Jeanette Grant. I head up the project management department at Adicet Bio. Adicet is, and I'll go into this here, still considered a startup, although we're now at over 60 employees. We were founded in late 2015 by Aya Jakobowicz. Aya is kind of a pioneer in the cell therapy space. And as I said, we're going through a rapid growth phase. So when I joined at the end of 2017, I was employee number 20, and we're going all the way up beyond 60 today. And we're doing kind of a strategic hiring plan from early stage, and as we're moving closer to the clinic, building out our clinical team there. So we are heavily skewed at this point towards research lab-based employees, and these are the people who are using Benchling, and I'll talk a little bit about them. As I said, our pipeline is in oncology, so we're building hopefully a broad oncology pipeline in the allogeneic cell therapy space. For solid and liquid tumors, and we're using gamma delta T cells, and I'll go into a little bit about gamma delta T cells, how they're unique, how we think they are kind of be a life-saving technology for patients, and how the complexities of gamma deltas have actually benefited from Benchling to help us drive forward. We are moving towards manufacturing these cell therapies, and I'll get a little bit into our timeline coming up. And we have a strong IP portfolio, and again, having Benchling in place has helped us kind of build up our IP portfolio and have a stronger suiting there. So gamma delta T cells, I'm betting maybe one or two people beyond the two Adicet employees here have heard of gamma delta T cells. Most people know about alpha beta T cells. Those are kind of the famous guys. They are the bulk of the population of circulating T cells in your bodies right now. Gamma delta T cells make up about 0.5 to 1% of the circulating T cells in the population at any point. They do have many advantages. Number one being that they are not, they don't express MHC molecules and therefore would not be recognized from a healthy donor into a sick patient. They also have, again, sparing of healthy cells. They have an inherent recognition and elimination of tumor cells. They produce their own cytokines and chemokines, so can support themselves in circulation. Now there are quite a few other great things about them. I encourage you all to check out the Adicet website. So we are building, Adicet is a growing company, and we're trying to build a pipeline to support the company and give us a long-term future. Our first product that's moving forward, we're aiming for IND at the end of this year, is a CD20 targeted CAR T cell therapy in gamma deltas. And then we have a number of solid and liquid tumor targets moving forward from there. You can see we have a pretty aggressive pipeline, especially considering the size of our company right now. But as we're growing, we're hoping we're hiring the right employees and utilizing Benchling to support this kind of rapid growth and moving really aggressively towards the clinic for a number of targets. One of the kind of important proprietary pieces of patent that IP that Adicet holds is around our manufacturing process. This is a really unique manufacturing process. We take PBMCs from a healthy donor, could be you or I or anyone in this room. We extract their PBMCs, we activate them using a proprietary technology to expand the gamma delta T cells alone and leave the rest of the T cells alone in the culture. We engineer them with CAR for our first product or TCRs for future products. They get expanded in culture, and from that we can get what we believe will be hundreds of doses from one collection from a healthy donor, and that can go into, again, hundreds of patients. So project management at Adicet, I was brought in again, I said at the end of 2017 to take what was really a research-based company and drive things forward. And so I put up here one of the favorite sayings of our tech neighbor in Menlo Park: move fast and break things. So one of the things I saw really quickly when I got in, we had a ton of great work that was being done. The documentation of it, a little spotty. It was going to be difficult for us to move things forward from an IP standpoint and also from a regulatory standpoint, making sure that we could pull all that data in, put it into an IND. We were doing paper notebooks at that time. As I'm sure a lot of the other people who have brought in benchling were struggling with paper notebooks. We had inconsistent usage of those paper notebooks, and we also didn't have a good process for storage of electronic data. So all the data we're generating these days is electronic. It was stored sometimes on people's laptops, sometimes on a public drive, sometimes in the cloud somewhere, sometimes on the machine itself, and then never came out. So we didn't have a good process for that, and so the company saw, a really obvious need for implementing an ELN and a broader than an ELN system, a really good data management system. So we went through an ELN evaluation process. We narrowed it down to 3 companies that we brought in and did an on-site evaluation and a demo, and we ended up choosing Benchling in March of last year. So we're really just a year out from rollout. Oh, thanks, Michael. And we chose Benchling for a number of reasons, one of them being the user interface was very clean. The second being it had a really user-friendly system for not just documenting, what was happening in the ELN system, but also translating that into a bioregistry and an inventory system. And all the modules that Benchling was advertising at that time, they actually had available. Some of the competitors were saying, oh yeah, we're gonna have this great system, but we'll have it in a year. And that was not useful for us at that time. So once we selected Benchling put together a really great team to help us roll this out. And I will give props to Nishant and Michael especially for really making this go well. So really quickly, we did a platform setup in about a month. We rolled out the ELN and MoBio system to our users really quickly after that. Then we spent several more months setting up the bioregistry and inventory systems. So the key aspects, the things that were really important for us to get the ELN platform set up, was picking these little things that you think don't matter, but really did end up matter, figuring out how you want to structure your folders. Took a lot of time, and we ended up, I think, with a really good system. The other thing that was key and that we liked about Benchling was having a flexible permission structure. So we have both our site in Menlo Park, but we also have a wholly owned subsidiary in Israel. And we wanted to be able to kind of silo some of the pieces of information between those two groups. We also have contractors or consultants that we bring in, and again, we limit access to the whole system to some users to just have certain folders. So having that permission structure and flexibility has been really useful. Putting together notebook entry guidelines, very difficult. It's something we're still working on, but the templates feature has been really useful for this as well. And then setting up witnessing policies. So that was another thing Michael was really helpful in implementing, is that in our Menlo Park site, we had a single signer system that was really easy. In our Addisat Israel site, they wanted to have dual signing, so it would go to a first level manager and then a second level manager. And Michael figured out a really kind of ingenious way to make that work. The bioregistry and inventory modules, getting those set up took a little bit longer. One being we needed to identify what we actually wanted to capture of all the things that we generate. And you saw that we have a very complex manufacturing process. All of those pieces are made in-house. So we had to decide which of those we wanted to capture and what metadata for each of them needed to be captured as well. So that took a little bit of time. So identifying those entities, selecting the responsible person who is going to be responsible for working with Michael on doing all of the backlog and the backfill, and determining what schemas needed to be set up for each. So overall, implementation of Benchling has been a great success. In the ELN, we have 38 distinct projects we've set up. In that, we have almost 2,500 notebook entries, and this is in, like I said, 1 year. And we have 50 authors who are continuously using the ELN system. In the bioregistry and inventory, and this really didn't get fully rolled out until about August, September of last year, so we've had a little bit less time. But we've had 1,500 unique entities created within the bioregistry. We have 9 different entity types that we're capturing, so they have distinct schemas for each of those. And we're tracking physical inventory in 7 locations. But ultimately, what Benchling is going to allow Adicet to do is to move rapidly into the clinic with life-saving therapies and has set up the company for long-term success in the future. The last key things that are ongoing is we're trying to train our employees towards consistency. And by that I mean, as new employees come in, that they're being trained to use Benchling in the same way as the older employees, but that the older employees are using kind of the same formats, and that we're starting to use the template, so we're capturing the same key pieces of data throughout our ELN notebook entries and going into the bioregistry as well. What's been very useful with that is instead of me just doing all the training for our new employees, we're also continuing to use the Benchling tutorials that are available online, and the chat support system has been really great as well, really quick turnaround on that. The last thing that we're working on implementing right now is the workflows module. So our process development group, the folks who are creating that manufacturing process, are doing essentially iterations on the same basic process, but they're changing little pieces each time. This is something we thought about when we first brought Benchling on board. We weren't quite ready for it. And as we've kind of made these massive process development changes, we're trying to capture what the impact of those changes are. So it's been, because our process is so complex, it's been a complex setup, but we've actually just, I believe, completed finalizing the evaluation and the setup of it, and we're going to start rolling out workflows to our process development group in the coming weeks. So I wanted to thank the Benchling team. They've been really helpful all the way through this process, very supportive, especially for a growing kind of young company who have a lot of opinions and a lot of needs. They've been really great. So that's it for me. Okay, so I guess there's Q&A.
So just curious if, the workflows module, if that's something that only the process development group is using, and have you considered expanding it to even research part where, you could say everything's a workflow in the end? So just curious how you guys thought about that.
Yeah, that's a really great question. So our initial thought was, because we needed more urgently to capture the data from the process development team, essentially to enable our IND submission, we wanted to start with the process development group and also kind of work out some of the kinks in terms of the flexibility of the system and figuring out how we as users would be able to kind of manipulate that and to do pulling out data from the data warehouse in the end. But we do envision using, especially in our discovery group, who actually does the early stage process development, it would make a lot of sense for them to use it as well. We are also using kind of a subset of workflows to do some of the bioregistry and inventory. So when we're doing these kind of bulk productions of some materials, Michael has set us up in workflows to kind of push those things into the registry a little bit more rapidly and making it less onerous on the user.
With engineered cell therapies being so complex to manufacture, I'm wondering how you sort of think about the interface between the research, cellular immunology group, process development, and manufacturing, and how some of these tools that you've used sort of help enable and facilitate some of that cross-functional work.
Yeah, that's a great question. So as a company, we kind of pride ourselves on being very collaborative, and we have project teams around each of these that include not only the process development and analytical development, but also the earlier stage researchers who kind of contributed and have the background immunology knowledge to help move these things forward. And again, that was partially why we wanted to bring Benchling in, is to enable, not just this, internal communication sitting down in a meeting, but if someone wanted to say, hey, how was this antibody originally derived, and what were some of the earlier results, even if that employee is no longer there anymore, because Benchling is so searchable and trackable, that has really enabled us to kind of go and do some historic dives.
My own company has a lot of parallels, I think, to what you just described, and I wonder if you use any external CROs for any experiments, and if so, do you import any of that data into Benchling, or does that sort of sit outside of it?
That's a great question. We haven't yet, but we will be soon. And that's something we should consider. Thank you.